So, hi everyone, I'm Natalia.
Hi everyone, and I am Jeannie, and we are the Marine Lab Research Assistants here at Point Blue Conservation Science. Yes, that's very true. Thanks, Heather. And so here at Point Blue, our mission is to advance the conservation of birds, other wildlife, and ecosystems through science, partnership, and outreach. So whether you aspire to be a marine scientist yourself, or you are just want to learn about our ecosystems and our oceans, or our marine food webs, which is our specialty here in the lab, we welcome you and hope you will leave here knowing a little bit more about the diet of the California sea lion.
Yeah. So first we'll just say a little bit about ourselves. So I'm Natalia again. I graduated from the University of Massachusetts Amherst in Natural Resources Conservation, and I did a concentration in fisheries conservation. And since then, I've had a variety of different experiences in marine science roles and working in marine biology education. And I've also done fisheries management roles. But I've loved being part of the Point Blue team, and I hope to continue to gain experience in the marine science field, and hopefully I will build a career in fisheries management.
Yeah, and hi, I have a background in fisheries policy and bycatch mitigation in industrial fisheries. And I graduated from UC Santa Cruz with a degree in marine biology. I enjoy my work here at Point Blue a lot as well, and all the work we do to conserve our oceans. And I really hope to continue this line of work and pursue my passion of continuing to protect sharks and rays and make sustainable fishing efforts more widely accessible. So and so here in the lab, we process a variety of samples collected from the predators at the Farallon Islands, which is just off the San Francisco Bay coast. And we also collect a variety of zooplankton samples on these very large research vessels with lots of scientists, and so we collect these like waters or plankton samples from the ocean, and that is how we go through like 6 or 7 different types of samples in the lab, and we examine predator diet samples to see what prey are most abundant at any given time in the ocean. And so today we're going to be focusing on the samples we collect from the Farallon Islands, the sea lion scat samples.
Yes. This is a picture of a California sea lion. So we look at the poop of these animals. California sea lions hunt a number of prey species, and they are opportunistic predators. So basically that just kind of means that they're not picky, and they'll take advantage of anything that is available to them to eat. The most popular prey reflected in our Southeast Farallon Island sea lion diet data are northern anchovy and cephalopods. So this is a northern anchovy. It's a little dried up, but they eat these guys a lot. And they also eat cephalopods. So cephalopods could include squids and octopuses, but most of the time in the samples we find squid beaks. So this is a squid, and the beak is right in there. It's like their mouth part. Here's a better close-up of the squid beak. But these are the structures that we find in the samples, in the scat samples, that help us determine that there were squid in the samples. So between the two, northern anchovy is of significantly higher nutritional value. It has a high fat content, and so it breaks down into more energy for our sea lions. And although northern anchovy and squid are the most dominant options that we see in these samples, we also find evidence of Pacific hake. So they'll eat a bunch of different types of things. So this is a Pacific hake. So they'll eat these guys, and they will also eat rockfish. That's kind of what rockfish look like. There's a lot of different species of rockfish, and these are found off the coast of Northern California here. And they also eat some species of flatfish. Yeah, so that's what we found.
Yeah, and so as you can see, they eat a variety of different things. And so this is why studying sea lion scat samples, or at least going through them, is really important, because by identifying what species of fish, squid, and other prey they consume, we can determine what prey species are most abundant and draw conclusions about the population sizes and the overall health of the ocean. We can also use this data to see how big events like climate change, El Nino, and other events affect the ocean's composition and how that ultimately affects the entire food web of the area.
Yes. And yes, that is the correct spelling of hake, Pacific hake. Yeah, so if you guys saw our preview of today's live, you would have seen the steps that we go through to process our sea lion scat samples. But if you missed it, we'll do a quick recap now. So let's go on a little tour and we'll show you what we do.
Yes. Alrighty, and we are just going to flip this camera around. Okay. Alrighty, so let's follow Natalia.
Come on,
and see where she takes us.
All right. So this is our freezer. This is where we keep all of our samples that we go through. I'll show you some. Okay. It's very chilly in there. So these are our sea lion scat samples that we're currently going through. We have a lot more in the freezer. But this is what they look like. It's really just a bag of poop that our scientists on the Farallon Islands collect for us. So yeah, this is what we process and look at. And I'll put this back in the freezer. And then I'll bring you over here. Of course, the freezer starts going when I start talking, but hopefully you guys can hear okay. This is our sea lion scat sampling or processing station. So what we do is we take that bag of poop and we put the poop in this mesh bag. So then we close the mesh bag up and we put it in this portable washing machine. So we let it run for a few cycles, and then what that does is it kind of washes away all the poopy particles that we just don't really have any use for, and then we're left with a bag of bones. So we'll see lots of different fish bones and any of those hard structures that are kind of harder to digest, and that's why they come out in the poop. And it's also what helps us identify what we're looking at. So we'll see bones, otoliths, squid beaks, and fish scales too.
Yes.
Oh, I'll put that back.
And these are all the things that we use to process our sample. And so we also use this big sieve to go through everything as well. And that way we can put our scat sample out of the bag into that sieve and then into a dish. And now we will take you back to our microscope. And we have a scat sample prepared for you already. So these are the dishes that we put, that we transfer our bag of bones into. And feel free to ask us any questions, because I'm going to be setting up the camera onto the microscope. So this takes a little bit of patience.
Ooh, it's a nice picture.
Yeah. Maybe not. Okay.
Has anyone ever used a microscope before? And if so, what have you looked at? I think the volume just went down.
Oh, yeah. Okay.
Yeah, let us know in the comments if you've used a microscope, if you looked at anything cool.
Hold on. I was having some volume issues. Okay.
Yep, ichthyology class.
Yeah.
Reminds me of ichthyology class too.
Yeah. Theology is really cool. Okay. So now we are going to be going through, so this is our sea lion scat sample. So this is what we look at under the microscope. And so as you can see, there's a lot of stuff in here. It's mostly bones. There's still some sea lion waste left over that didn't escape. So it can get pretty smelly. But if we just go through it and if we let it soak in alcohol, that usually minimizes the smell. But yeah, we have a whole bunch of stuff. This is a really colorful rock over here. And so yeah, so our job, so this will take us like a day to go through because we got to go through every little thing. And then we find scales that are like this and bones like that. And so what we find are fish bones. So these are miscellaneous fish bones. Yeah, there might be cartilage. I don't know if cartilage gets digested or not.
I feel like I have seen pieces of cartilage. They're kind of like squishier than the other pieces, but I can't be sure if they're cartilage or not.
Yeah. Yeah, so, ooh, whoopsies.
Nice.
Yeah.
Yeah.
But we definitely see cartilage, and these are just miscellaneous fish bones that we can't really use to identify. And then we also find lots of vertebrae. So we process in a season, how many sea lion samples do we process in a season?
In a season? Well, I can tell you right now, for July of 2022, we still have 12 left to do.
Yeah.
So we do have quite a few. I don't know. Maybe Meredith has a better answer to that one.
Yeah. I think we usually have like 20 or 30. But yeah, these are vertebrae, and if you can see the little holes in them, like right here, that little hole, that's where the notochord goes. Okay, yeah, so yeah, we have about 25 samples per quarter. Thanks, Meredith.
Yeah.
But yeah, so we find cool vertebrae, we find cool fish bones, and then the fish also leave behind these little fish eye lenses. And so these are just like hard little balls. They're like these golden little balls, and we just find lots of them in our other predator samples as well. So they're pretty cool. So yeah, we'll like go through a whole sample in like one day because it's like a giant pile of bones. And then once we go through it, yeah, there are certain things we look for. So we pick out,
Yeah. Oh, Meredith says we go through approximately 100 sea lion samples per year. That's a lot.
Mm-hmm.
That is a lot of poop that we're looking at. Yeah, I can zoom in. Yeah, so these are one of the structures that we look at. These are northern anchovy scales. So the most common types of scale, let me see if I can. Yeah, the most common types of scales are cycloid scales and ctenoid scales. So anchovy have cycloid scales. They are so pretty, Lishka, I know. We find a lot of them. Yeah, so they're kind of thin and flimsy. And I feel like when most people think of scales, they're thinking of ctenoid scales, which are the ones that have like the rings. And so they look kind of like a fingerprint. Fish like rockfish have those types of scales, but anchovy have these ones. And we do find rockfish scales sometimes in these samples, but not in this one. This one had a bunch of anchovy scales. Another thing that we find is otoliths. So these are also northern anchovy otoliths. Let me see if I can get a better. Oh, there we go. Yeah. Yeah, so these are northern anchovy otoliths. What is an otolith? An otolith is fish's inner ear bone. So fish have two main otoliths. So they'll have one on each side of the head, and we can actually tell what side of the head an otolith came from. Usually if it's not too eroded or anything like that, we can tell that, and we can also calculate. So the otoliths, we measure the otoliths, and then we can use that measurement to calculate the size of the fish that it came from using a formula. And so ours are calculated in our database, so it calculates that for us. So yeah, the otoliths are pretty important too, because since we can tell what side of the head the otolith came from, we can count how many fish are in that sample. So it gives us kind of a good idea of the species of fish and how many are in that sample. So those are northern anchovy. I also have, these did not come from this particular sample, but these are Pacific hake otoliths. They're a bit bigger. They're not always bigger, just these particular ones are bigger than those particular anchovy ones. Yeah, pretty cool.
Yeah.
I'll let Jeannie take over.
Yeah, and so then obviously this sample that we are going through right here, there's a lot of evidence for fish. So there's like a lot of fish bones and fish scales, and so obviously, you know, this sea lion sample had a really a strong preference for northern anchovy, but sometimes when northern anchovy aren't available, they also prefer squid. So we found these squid beaks from an earlier sample, and if any of you follow me, can anyone guess how many squid the sea lion ate? Because sometimes it varies, but this one we found a lot. So and the way we know how many squid the sea lion ate is because each squid has a top beak, which is this one, which is very pointy and narrow. And Leash guessed 500. That's a lot. That's a good guess. But it's about a tenth of that. So it's about 50 squid that this sea lion had eaten for breakfast. Yeah, 501. So yeah, so there's a top beak, and this is a bottom beak, so it's wider and shorter, and it has some ridges on it. If you can see, it's like not as smooth. But yeah, so we count the top and bottom beaks, and so we can sort of pair them up. So if we find like 10 top beaks and 8 bottom beaks, we at least know that it ate at least 8 squids. And so in this sample, we actually found 51 top beaks and 51 bottom beaks. So it perfectly ate 51 squid, which is pretty crazy. And this is actually, let's see, I don't think I can show you guys. But yeah, but it was a lot. So it had 102 squid beaks in there, which is really crazy. But yeah, and so it's important for us to go through these samples so that we can see, you know, what they're eating, and like, why would it prefer squid when, you know, northern anchovy are really nutritious. So yeah, so it's just interesting to see. And now we are going to switch back. Yes. So this is our lab, and just 1 second. Alrighty, hi everyone. So yeah, so once we're done going through our microscope, and once we're done like processing and going through this entire pile of bones. If you recall, the 3 things that we extract from that pile of bones are otoliths, scales, and squid beaks. We then have to store those separately, so that way we can store it for our data in case we need to go back for any reason. So yeah.
So this is what it looks like. So we put all our important structures. It's just that anchovy are very nutritious. They're very fatty, which we like. So yeah. So we have our vials of scales, and there should be some beaks in there as well. That's where we keep them. This is the sample with the 51 beaks. I don't know if you can see tons of beaks in there.
Yeah.
But and then our otoliths, we keep in these gel capsules. Usually they're pretty small and they can fit in the gel capsules. And so each gel capsule is one sample of otoliths. So that's where we keep our samples for the future.
Yeah, and so obviously this work is really important. And as I said earlier, we have been collecting these sea lion scat samples since about 2014. So we have about 8 years of data. So we started December 2014. And this is really important because it helps us see if any big events happen. So I don't know if you guys are familiar, but there is a thing called the Big Blob, which was a heat wave that went across the coast of California or across the ocean in the Pacific, and it actually wiped out a lot of the kelp forests because the kelp forests thrive in colder water, and it destroyed a lot of the ecosystems and caused a huge disruption. And so with that big blob, we noticed in our sea lion scat samples that there was a huge decline in the number of anchovy they were eating. And they were mostly eating other fish, and squid was making up a large portion of their diets. And we didn't really see northern anchovy come back into sea lion diets until the end of 2018, beginning of 2019. So that is why we keep all our data stored in these vials, and why we record all our data and record all the species that they eat, and we upload it into the Point Blue database cloud. That way we can always look back on it. And so, so I think that is just about it for today. Does anyone have any questions?
How long do we keep the samples for? I think Meredith can answer better, but I think it's kind of indefinitely right now.
Yeah, we have a lot of boxes of samples all over the lab.
Yeah. And these are extracted from sea lion poop.
Yeah, so if you watch our TikTok that I made, that sort of shows you the process of how we clean the sea lion poop, and we're just left with the bag of bones that we go through.
Okay, Meredith said we've saved all the analyzed samples going back to 2014, so since then, yeah. Awesome. Oh, thanks, Heather.
Yeah. So thank you so much to everyone who dropped by to visit us today. We hope you learned something new and were inspired and had a lot of fun with us, because I had a lot of fun with you guys too. And so if you would like to get more involved or even volunteer here at the lab or volunteer one of the other departments at Point Blue, we have volunteers for the Farallon Islands right now if you want to collect sea lion poop with them. Feel free to reach out to us or check out our website at pointblue.org. And if you feel so inclined to make a donation, we would really appreciate it. And so make sure to follow us on Facebook, Twitter, Instagram, and we will be posting a recording of this on YouTube as well. So please stay tuned, because we have more lives coming up in the next few months. So on February 14th, we have a native plant nursery live on Wednesday, February 14th. So be sure to drop by for that. And then we will also be doing another live from the lab on Wednesday, March 13th. And we will be going through a seabird pellet sample. So that's another big predator for the Farallon Islands. So thank you everyone for watching, and we'll see you next time.
Yes, thank you.
Thanks.
Thank you guys.
Bye.
Thanks, Mom, for joining. My mom joined.
Oh, really? Oh.
Hi, Delisa.
Thanks everyone for joining. Let me know if you guys have any questions, and feel free to reach out.
Yes.